Wait, Parama? Okay, Parama is actually a student who just asked me about adding to the class.
Oh, well, welcome, Parama. I was like, I don't recognize your name.
Um, also, I
You just lost your connection.
Hey, can you
Yep.
guys hear me and see me now?
Now I can hear you, yeah.
Okay. Um, yeah, I go by Pia.
Pia, P-I-A.
Yeah.
Okay, sounds great. So Pia, every aspect of the class has been recorded and is available through the webpage. So yeah, you're
Yeah.
welcome to add it. I think there's still plenty of time to catch up.
Okay, that's awesome.
Good.
I guess I just need to make friends now.
That's great. Perfect. So the way that you can do that is everybody has recorded a profile. Daman will be sending you all of the information about how to access those.
Okay.
Okay. Let me put two links in the chat. So one is the feedback and one is the instructions.
Okay.
So, okay. So for all of the rest of the folks who have written their top 3 ideas, can you please raise your hand either, on video or through the Zoom channel to basically say if you have not received feedback on your project, top 3 ideas. Then we'll do that now first. So has anybody not received feedback? Oh, Thomas. Okay, let me go through your okay, perfect. All right, so I'm going to share my screen and then go through feedback. And if anyone else hasn't received their feedback, just type your name in the chat window, and then we'll do that now. All right, so Tom, based on the scenic paper, implement the suggestions for scaling to larger cRNA datasets. For instance, advances in gradient boosting could be implemented to speed up determination of co-expression of genes. Specific steps in the workflow could be tinkered with. A different method of measuring the activity of a gene used. Scoring would be done. So I feel that this is more of a bioinformatics project, challenging aspects, but unfortunately, the biology is, less enticing since it's just about speeding up an existing algorithm. If you can show that the speed up allows for, different types of applications, or perhaps a super interesting outer loop, that'd be great. Okay, so Thomas, does that make sense? Can you hear me?
Yeah, that makes sense.
Okay, perfect. So yeah, I mean, if the speed up is sort of achieving something that's like qualitatively different, that'd be great. But if it's just a quantitative speed up, it's not as exciting. All right, idea number 2. Create a model that can, but again, this is the type of component that would be super in a team project. So if there's a team project that this can be a part of, that sounds fantastic. So you can look for people that have something similar and then see if you can sort of contribute towards that direction. Generate predictions of phylogeny, either at the species level or the DNA level. Yeah. For specific species, group of species, molecular, trained on past phylogenies, biological configuration, undergo rapid changes, most useful to testing and scoring. Certain papers already go into this route. This paper predicts phenotype from genotype. This paper creates a model that predicts evolution in E. coli. The idea would be to create a general model, but this seems inherently difficult. Thus, focus on a single species strain, coronavirus. However, it would be possible to create a general model, predictor selection drift, other interesting aspects. Yeah, so all sounds fantastic, but how do you test whether you're right or not? Right? So basically, you need the right benchmark data set. So basically for every single aspect, if you're trying to sort of find selection versus drift, you have to find a place where it's annotated. And then you can go back and sort of see if you can predict it right. So basically with evolution, the challenge is that evaluation is hard because there's few sort of gold standard data sets. But if you find one, then that sounds like the right project. Does that make sense, Thomas?
That makes sense.
Evolutionary pressures and mosaicism. Oh, cool. In the context of cancer cells and the host and different genetic conflict. I love it. Oh, that sounds fantastic. Strong effect mutations, low frequency mosaicism allows them to rise in frequency, determine which mutations are disease causing, also be drawn. Very, very cool ideas. We have some results on mosaicism in Alzheimer's that you could build upon. Talk to Carlos lab. He can point you to data. And then Jackie Yang has mosaicism in cancer single cell RNA-seq. So you can talk to her as well. So basically, this, even though that's the one you were most concerned about, this is actually one where there is data. Okay? And then she had some cool results in mosaicism already, and you could totally take that on. So I think this seems, most promising. Okay, so contact her and then take it from there. Sounds good?
Yeah, thanks.
Right. Who else hasn't? So Michael, okay, and Hugh, perfect. Oh, and Reina. So let's do Reina first.
Manoli, before you go ahead, can I ask what we are supposed to do in this mentoring session?
So the original goal was, if we have a sufficient number of people, was to sort of find people who do not have teams yet, and then use that as a way to match up. So Daman, here's what I can do. I can make you the host, and then we could, so let's do the following. So basically, I'll go in a breakout session with the people who want one-on-one feedback. And then what we're going to do first is I'm going to create the following rooms. So basically, room A or room 1 is going to be Manolis plus one-on-one feedback. Okay. Now, what I would love to have is proposals for what topic you guys would like to cover for the other breakouts. So basically, for every breakout, we can basically create topics that folks are interested in chatting with. So basically, there were a lot of people on single cell. So I would say room 2 is single cell. Speak up for the other rooms, and let's see how many other folks. So basically, who's interested in joining? Can I ask everybody to turn on your video so I can see your hands as you're raising your hand? So basically, who's interested in joining the single cell breakout? So, okay, we have 1 person, 2, 3, 4. Okay, perfect. So then we are having a single cell breakout. So for teammates, I was thinking of doing the teammate matchmaking per topic rather than sort of having a single room for all of the teammate finding. Daman, do you agree with that?
Yeah, I think that's good. I think it would be super helpful if if everyone could rename themselves to whatever room they want to go in, and then I can just put everyone in their respective rooms.
Yeah. So
just add it to your name.
Yeah, for the single cell folks, just rename yourselves already. And then let me create, oh, I'm not the host. Are you the host, Daman? You're muted.
Yeah, but I will make you host. Okay, you should be the host now.
Yeah, yeah. So I'm going to create, I don't know, maybe 8 rooms so that we have them as we need them. And then for folks who have renamed themselves, you know, so breakout room is going to be one-on-one. And then 2 is going to be single cell. And then, Daman, I'm going to make you the host again so you can do all that. And then I'm going to go to the breakout. So basically, what other topics would you guys like to have for additional breakouts? So, okay, awesome. I see people who are already naming themselves room 1. So I'm assigning you to those. And then room 2, I'm assigning you there. So single cell, single cell.
There's some suggestions for other topics in the chat.
Yeah, great. Predictive modeling, graphs, interactomes. I like that. So I'm going to call room 3 networks /graphs/interactomes. So if you guys would like to go into room 3, just write room 3. And then predictive modeling, I guess room 4 is going to be ML. But ideally, we'd like the ML predictive modeling. 3 networks, graphs.
Can we go between the rooms?
Yeah, you could go to multiple rooms and then just basically come back out. You can say leave room, and then you can go there.
Yeah, I can reassign you. Just let me know.
So I see some people for room 3. I see some people for 4. Some people for.
Professor Kels, I could take care of this if you just want to go and do your one-on-one feedback. And
I've opened all the rooms, and then room 3. Room 4. Okay, so basically, Daman, I'll let you. I'm making the host.
Okay.
Then I'll go into the breakout for 1, and then whoever else wants to join 1, just come to 1. Okay, so can you assign me to room 1? Because I don't see how I can go there.
Yeah.
Okay, awesome. This is fun. Hey guys. This is awesome, multitasking. In real life, it's actually harder to do this, but now it is easy. So here you were next, right? Oh no, we had Raina, but she's not here. So let's do here. All right, using single cell chip-seq data. So SC attack more common to reveal heterogeneous chromatin states in tumor cell. Beautiful, I love it. Endpoint. Okay, so we have SC attack data from cancer immunotherapy across both immune and tumor cells. Dr. Jackie. How cell adapts to the environment by changing epigenetic mechanisms. According to this paper, cRNA-seq is used in parallel analysis. Different ML models can be incorporated. But we also have different ML models can be incorporated to predict drug response in cancer cell line. Ah, very cool. See, CCLE and project Achilles. Code. Okay. Possibly combine with small molecule response perturbations for different melanoma cell lines. So does that make sense here?
I'm not sure like how to combine with the response perturbation.
So basically the idea there would be that you basically can, so you have the single cell RNA-seq data and you basically build clusters of cell type specific responses, sort of see how responders versus non-responders change. So find differentially expressed genes during response. Then from CCLE data, see which genes change in expression in response to which drugs. Then match up expression profiles between drugs and cells. Okay? Does that make sense?
Yeah, that makes sense.
Cool. All right, idea 2, using sRNA-seq to identify differential network. Recently, there's a lot of new tools to infer the differential cells. We can use these tools on desired sRNA-seq and use the results to give insights into cancer. I love it. Also take ideas behind these tools. Yeah, fully agree. Great area, lots of data sets, challenge, make it specific, have a hypothesis area, and find data sets for validation. Does that make sense? So just, beware of all of that. Number 3, identify variants associated with specific disease. Using RNA-seq, ChIP-seq to annotate the functional variants, gene function based on functional variants. So this is a very broad area, lots of data sets. See our recent IPMAP paper on this. Is that okay? Sounds good. Okay, awesome. So I think Raina, you had entered your name first in the other room before the breakout. So let's go through yours.
Thank you.
All right. Apply cell lineage mapping, tracing to AML, sRNA-seq, as they found large amounts of heterogeneity. Interesting. Yep. Sounds great. Make sure you have rich enough data set with validation. AML may be too easy data set. But overall, it's a great area. I think you just have to find, specify, build hypotheses, find data sets for, cross comparison. Does that make sense right now?
Yeah, or what was, so cross comparison, you mean like other data sets to like validate whatever pseudo-time outputs, right?
Correct. Basically, you have to have some orthogonal data. So it could be coterms, ideally higher resolution molecular mutations or something like that. Yeah.
Okay, gotcha.
Okay, so use cell clonal reconstruction methods to remove neutral tail mutations I'm not sure I know what neutral tail mutations are.
It was in the paper that I read for this class, they identified that like a lot of subclonal reconstructions will look at like mutations that just occur from
Yeah.
like cancer cells dividing and use that to define clusters, even if they're not truly like driving division.
Non-drivers, basically.
Yeah.
And see how that affects clustering of AML. Single cell data sets, and if that affects, describe how the removal of neutral tailed increases the accuracy. Yeah. So here's the challenge. Subclones need neutral to identify. Drivers need driver mutations. So basically, the advantage of having a bunch of neutral mutations is that they allow you to do the phylogeny. So actually having the neutral ones helps for phylogeny, but then you need to find the driver ones. Do you see what I'm getting at?
Yes, thank you.
Yeah. But okay. Compare cell lineages and clustering of AML samples, or maybe immune cell samples. Cell lineages and clustering of AML samples, immune cell samples from the same patient over time. See how clustering and differentiation change with respect to chemo treatment. Perfect. So Jackie Yang has scRNA during cancer immunotherapy response. Yeah, so then, sounds good. I think that's your email, but I'm not sure.
Gotcha. Thank you.
Oh no, that's not it. Jackie Yang. Okay. Sounds good. So you can leave the room and come back and to the single cell. Okay, so Michael.
Thank you.
I think I'm like all the way at the bottom. Oh, you found it. Never mind.
Yeah, it's mouse aging transcriptomic atlas, CRNA-seq in a variety of organs and tissues, different time points. Same group generated complementary. Complementary means it's free with your room. Bulk RNA-seq data, for instance. Built using ML and aging signature, both within and across cell types. Rate of aging. Talk to Zena, my group. Yeah, sure, I can see that. Leverage the fact that the data was collected from multiple organ systems of the same individual mouse. So see GTEx data in humans for this multi-tissue collection.
Yeah, I'm familiar with the GTEx project. It's basically the human equivalent.
Yeah. So, you can have aging as the phenotype.
Yeah, one benefit I was thinking of using this mouse cell atlas is that the actual genome is the same and the environment is the same, so it might be easier to zone in on aging factors, but I don't really know. Yeah, all right.
Yeah. Possibly combine human and mouse and look for similarities and differences. Okay. and single-cell RNA can bring sample from human chip bonobos and macaques. Now it's focused on patterns of expression across species, cell types, and brain regions. Difference in the bulk and single cell. Yeah.
What is orthology?
Basically, orthology is finding matching genes across species. It would be nice if you had the same gene name in all of the species, but no, you have to use phylogenetic. Okay, so let me leave the room and I'll try to stop the stopping. You have to explain all the rooms.
I think I lost host capabilities.
I see. So let me make you the host. I made you the host. Okay, so then you can.
Should I close all the rooms?
No. On the contrary, keep them open.
Oh, they're open.
Okay, but they were closing. They close automatically after 15 minutes.
Oh, okay.
You want to put me back in room 1?
Yeah.
Oh, yeah. Sorry, guys. I just went to sort of prevent the closing. It's like when you know, the super user has to go back out into the matrix and then come back in. Or something.
It's exactly like that.
Exactly, just like that. Yeah, so I think it's a great project. Sounds good?
Yeah.
Okay.
Yeah, it's a super cool data set.
Yeah.
Yeah.
Hypoxia, low oxygen, shown to have clinical relevance for people suffering from metabolic disorders. RNA-seq data for a variety of tissues. Here would be cool to use ML to better characterize the hypoxic response. Could be interesting to compare our results from these hypoxia-related CRISPR screens. Yeah, so I would focus matches between fish and human. Does that make sense? So if you can have sort of a fully human contained data set and analysis, I would sort of focus on that.
Yeah, would it also be fine to just focus on fish, because I couldn't find any hypoxia related human data sets. I mean, that was also my preference. I just wasn't able to find it.
Yeah.
There's human cell lines, but I preferred the full organism fish over just like human cell culture.
Yeah, but you have to match with human. You have to. I'm sorry. It's I feel that your relevance is going to be smaller otherwise.
Okay.
Yeah, but I think ideas number 1 and 2 are great already. So
awesome. And then would you mind, actually, Audrey, who's partnering with, is also here. We had a question about one of her ideas, which you've already commented on. We're thinking about working with Na. Audrey, actually, would you mind telling us more about like the general situation you've been coordinating?
Yeah, sure. So I think about a week ago, Na reached out to about idea 2, and we now know that she's going to work on a different project, but she's also still interested in potentially collaborating a little bit with us on this project. She's interested in perhaps using our results and extend our results for one of her projects that she's currently working on at your lab, I think.
sounds great. Sounds great. I'm delighted.
Yeah. Yeah, we were wondering if it'd be okay to just be a group of 2 with a little bit of help from Na, even though we know it's kind of frowned upon not to be 3, because Na's going to be pretty absorbed in her own project.
My guess is that there's so many people interested in aging and single cell stuff that you will probably find someone. Like if you just search aging, I'm sure you'll find multiple people who are interested in that. So basically there's Audrey, there's Mildred, who's also interested in aging. Right?
Oh yeah, so we'll look for a third and then kind of collaborate with Na on the side.
Okay, that's what I think.
Okay.
Okay.
That sounds reasonable. Yeah.
Okay. Perfect. All right. Thanks, guys. Bye-bye.
You also never like commented on any of my ideas. I don't know why. Can you like go through them?
Yeah, of course. I'm going through them now. So
also, I don't know, like, is there any list of people who still don't have partners? Because I really don't know how I'm supposed to do that. Like, if there would be some kind of list to like email people.
yeah, so I would basically go through people's projects and profiles and just send them an email and sort of look for matches.
Yeah, but like I don't know which of the three projects they choose and whether they already have partners. Like maybe they already have two partners and they don't need a fourth person. Maybe they like.
Yeah. So yeah, that's a good point. So I would you know, sort of go back to the team building sessions as soon as possible to sort of try to match. But I think most of the people don't yet have. I mean, my guess is that a lot of the people still don't have finalized teams.
Okay.
All right. So interim life evolution in eukaryotic evolutionary tree. So look at papers in the area. This was, you know, hotter a few years ago.
I didn't find any on like Intron lands. I think there are pretty good ones on like Intron gain loss, but not on Intron lands change. And I don't know, like most of them I'll have like pretty narrow scope. Something like we take like I don't know, one genus of like those fungi or like those animals or whatever, and like analyze through them, or like we look at one gene across like many species, but maybe that's like trend specific to the gene. And like the idea was that you could like do it to find trends and like
Yeah. So yeah,
wait, what the hell does it have to do with
what will be the biological insight is my question. So basically, you know, how relevant will these be? Will people care? So, I mean, it's, you know, it's gonna be a challenge. So codon gain loss in other eukaryotic lineages using algorithms similar to the one. Sorry, the Kunin article. Codon gain loss. What do you mean codon gain loss? Like basically.
Fuck, no, I meant Intron gain loss, something like, it was basically to do the same thing, but like with more species. But I think that's a bad idea now, because like, I think that size already done, like it's not that interesting. Okay.
Diversity, ecological conditions, taking field mission. Oh, I remember. I went through those before. That's why I didn't give you feedback, because I found them all to be kind of difficult to execute. And I remember I actually went through them, and I was like, it's difficult.
That makes sense.
Yeah. So
Probably
basically,
I should just
I
join someone doing something more realistic.
and not as rich an area. So basically, my general advice.
What do you mean not a solution area?
Hold on one second. Let me just dictate my thing. So my general advice to you would be to team up with one of the other teams that are interested in cross-species conservation between human, mouse, zebrafish, and other such species, because you will have a good set of partners and you will use your interest in evolution to the maximum while finding partners already. I think if you focus on trying to get a new proposal in the pot, you will have a hard time finding partners for it, period. So I would instead focus on reading through the existing ideas for the keywords evolution and something along these lines.
Makes sense.
Okay. Does that make sense?
Yes.
Okay, let's go back up.
Yeah.
Sounds good.
I think it's always been the case that something pops up.
Hi, Dom. Did you want me to talk to any of the folks here? Or should I go into any of the other rooms and see if they need help?
I think everyone here is just like either like just in a transition.
Oh, there's a join breakout room. I'm so like I did not see this tab. Thanks.
Dama, can you make me the host again?
Yeah.
So I can go and see the different rooms, and then I'll just jump between rooms.
Okay, I will not have control over anything.
Yeah, can I give you, no, I'll jump between rooms, but then I'll,
Okay. All yours.
Okay, so where do you want me to put you?
You can put me wherever, anywhere you want.
Or you can stay here and basically.
Yeah, that might be good because people are moving back and forth rooms.
Yeah. Then why don't you just put me in the single cell room?
Okay.
And when I come back out, I'll ask you to put me in the ML. Actually, no, put me in the ML. That's probably the place that has the most room for input.
Okay.
Okay, so I'll just make you the host.
What rooms do exist right now?
And put me in room 4 if you can.
Okay, done. Tatiana, all the rooms there's like single cell, networks, and ML.
What is okay, what is machine learning?
So Tatiana, my suggestion would be to search through and find partners and just email them right now. You have a very specific interest in evolution, and then there's only very few groups that are interested in evolution. So reach out to them would be my suggestion.
Okay, just hard because I don't know. It's very long list, and it's not sorted in any way.
Yeah, but you can use the search function. You can use Control F to find stuff, and then search for evolution, mouse, etc.
Or you can make a Piazza post, Tatiana, and just say, "This is what I want to work on." Like if anyone wants to.
Yeah. Or just send a message basically saying, I'm interested in joining a group on evolution. Daman, I don't think I'm assigned to the breakouts. Oh, yeah, James.
It looked like it was promising.
You can probably just ask.
Oh, Manolis is here.
Hey,
Yay.
I'm interested in single cell. Anybody has any project ideas I could join? So are you guys partly in teams already? Lily, I know you wanted to chat with me at some point. Do you want to go back to the main and then go into another breakout together and talk?
Yeah, that would be fantastic.
Okay, why don't we both leave and then we can go talk. Okay, sorry guys, I'm only transient. Leave room.
I can't hear you, Professor Callis.
Can you put me and Lily in group 5? She wanted to ask me some questions about partners and stuff.
Sure.
Perfect. Like I'm calling the Matrix. Operator, I need a room. I need
Okay,
it.
that should be good. You should be able to go.
Okay, sounds good. Let's join breakout room. So you have to click on more, join breakout room, Lily.
Okay.
Your question sounded pretty confidential, so I want to make sure that you have a chance to ask it. That one?
Yeah, idea one.
Okay. This sounds like a super totally uber, technical project. So basically one idea could use TFs for gene regulatory modeling. See next lecture. Epigenomics. No, not the next lecture, but the lecture after. Yeah. So
So basically what they're trying to do is predict if you see like a bunch of like a series, what the next nucleotide is going to be.
yeah, but this is so ill-posed. Basically, I think that you can definitely improve it dramatically by sort of thinking about, what does that even mean? And, I think that the challenge is, your part is going to be the most complicated one, basically, in figuring out how to even guide these architectures. I think having people who can sort of really think about those, in an NLP kind of fashion. Just a sec. So lecture. Basically, go use NLP to infer parameters or that would be my suggestion. So you can look at that lecture for insights, basically.
Okay.
There's okay. Does that help?
Yeah, that helps a lot because then my other option was I was trying to set up a meeting with them, but they're both on European time, so it was a little difficult. So then I was like, I'm just going to reach out to some other people in case this doesn't work out, because I talked to Joanna today and she told me that they were actually talking to another bio-focused person and they wanted 4 people in their group.
Yeah,
which I don't know.
I think I can allow 4 people.
Okay. But I also talked to Sonia because she's also interested in predictive modeling. And she looked like she had a very strong like computer science and biology background because she has like bachelor's of science in both biomedical engineering and computer science. So she was more interested in doing the predictive modeling to find the most suitable treatment or medicine or whatever based on single cell RNA sequencing, because if you know the pain medications that work well with specific.
I think that's a project that's more likely to have a good, strong biological component. I think the other one is very, very technical, and you might just really have a hard time translating to biology.
Yeah.
Yeah, so I think the last one you just mentioned sounds like the most plausible one.
Okay, because I was like looking at Ricard's project and I was like, this is like a lot of algorithm and not a lot of like biology.
Yeah, no, I know, and that's a challenge. And I don't think I mean, basically, you'd have to be a magician to add a lot of biology to that, and it's going to be difficult.
Okay.
Yeah. Although
So-
working at the motif, grammar stage, so look at that lecture and sort of look for ideas through that.
Okay. Because I think Sonia was really interested in working on any of these 3 projects. And I looked through to try and find some data sets, and there are a lot of data sets actually that have single cell RNA and have the corresponding patient like results available for like different types of cancers, for multiple myeloma, for Alzheimer's. They already have predictive models based on them because they're predictive model papers. But what we were talking about in the breakout room was that like a lot of models that are made aren't necessarily good models. They can like change the numbers to make them look better, but it may not be like the best or most efficient model.
I agree, completely agree.
So would that be like a good way to go? Or because I was just like had 2 different options and I didn't know how to weigh them, and I didn't want to go down one track and then like 2 weeks from now be like, oh, I really should have done the other thing. So I figured your advice was going to be like really important for this.
Yeah. Yeah. So I mean, again, look into more details about the grammar one. You know, at the motif lectures. They both sound plausible. I just, you know, you would have to really steer the ML team to be, you know, much more focused on motifs than nucleotides. And this whole predicting the next, you know, basically predicting the next nucleotide / motif is nice, but really in the framework of an RNN which learns you know, time course relationships in data. So that would be my suggestion there. So basically, don't think of the predicting the next one as the goal. Nobody cares about that, but everybody cares about sort of, if you can build such a model, then what does that tell you about the temporal relationships or about the, you know, colocalization relationships of different motifs and factors. Do you see what I mean?
Yeah, focus on the relationships of the different like motifs that are available and why one would lead to the next.
What does the temporal relationship teach you about how these motifs function? But again, I find that project to be quite challenging. And I think at the end, it's going to be just a bunch of cool machine learning with very little biology. So I don't want you guys to get penalized because of that. So I don't want to put you in an impossible project, basically, where your role is to bring biology to a non-biologicable project. Do you see what I mean?
Yeah, so it may be better to go with the predictive modeling one. Okay.
I'm guessing. Okay.
Awesome. Thank you. This was so helpful.
Okay, good. I hope it helped. Talk to you soon. See you in the other room. Hey, Devon.
Okay.
Lily, where do you want to go next?
Can't hear you.
Lily, you're muted. Yeah.
Shoot, my bad. Sorry. I'll go back to breakout room 4.
Okay.
It's like a video game. I'll take door number 3 now.
Okay, you should be good to go, Lily.
I just click join room, like on the bottom right. Yep, she's gone. Cool. This is working out. Seems like people are chatting and all of that.
Good.
I love it. How does it feel to be the master?
It's fun. People came and like talked to me about their ideas and stuff too, so that's good.
Great. I love it. This is fantastic. All right, operator, can you put me in a random room? Which one has the fewest people? The networks one?
The networks is empty right now. I could put you in it's like everyone is in ML and then 2 people are in single cell. Do you want to talk to the single cell folks?
Yeah, I don't.
Go to ML.
Just put me in a single cell. That's fine.
Okay. There you go.
Great. Oh, I'm alone.
They must have just left.
By the time I joined, they were gone. So just put me in the other one.
Okay. There you go.
Great.
analysis with like gene data in, it was like chromosomal aneuploidy, but I have a lot of coding experience. I have some ML experience, and I think it's an interesting project.
And it seems like nicely scoped, which seems good.
Yeah.
Yeah. Um,
Hi guys, I'm here. Do you guys have any questions for me, or are you just all set chatting with each other and I can just go back up?
Oh, actually, we were wondering about something beforehand. What kind of computational resources might we have? Because neural networks and things like this are very like computationally intensive.
Yeah.
Might not be.
So last semester we had basically collab accounts that Google is sponsoring for students. If you talk to Brian, I think he can set something like that up. So email the staff, email 6047-staff, and put a comment on Piazza basically saying, hey, what are the computational resources? Professor Kellis mentioned possibly using Google Colab or some Google credits that similar to what we had in 6874. Is that an option?
Awesome. Thank you.
Perfect. Okay. Any other questions? Okay. I'm going to go back up. Bye, guys.
Bye.
Hello.
That I was remade the host for some reason, right?
Oh, I didn't do anything. I still have host powers.
Oh, okay. Sounds good. It's funny because when I clicked breakout rooms,
Mm-hmm.
kind of showed me the list of breakout rooms. So anyway, I guess I'm co-host, that's why.
Awesome.
Good. So that's the end of my, availability. So I have to run soon. Are there other issues we should talk about?
So the recitation recording from last week recorded to your cloud, it said.
Let me see if I can download it.
Download that, that'd be awesome.
What I usually do is that I do Alt-C, which basically stops the cloud recording, and then Alt-R, which starts local recording.
Okay.
So it automatically records as soon as you join to the cloud. So let me see if I can go find it.
So it would have been on the 18th last week.
September. You know what? Let me just click on any of them.
I think I saw an 18th on there, but
yeah, but let me. I think when I go here, I'll be able to just see them all.
Okay.
Yeah.
Oh, awesome.
Great. So my guess is that it's the big one, but there's two of them.
So it would have been the one at 3 p.m. I don't know if that's helpful.
Copy sharing information.
Yeah, I can just go through them and see which one it is.
Yeah, exactly. I'll share both of them.
Awesome.
My guess is that the other ones are nothing, basically.
Yeah.
But those two are the important ones. Anything else?
No, I think we're good.
Awesome. Good. Thanks, Aman.
No problem.
Bye-bye.